Hello everyone and welcome back. Have you ever thought about how a tiny mistake in our body's instruction manual, like a single typo in billions of letters, could lead to a lifetime of suffering? For millions of people worldwide living with inherited genetic diseases, it's a harsh reality. Today, we are diving deep into one such condition and the groundbreaking scientific achievement that's not just offering hope but literally rewriting life itself. We are talking about editing our very genes, our DNA, to potentially cure diseases once thought incurable. Are you ready for this incredible story? We are talking about sickle cell disease, also known as sickle cell anemia. It's an inherited disorder traced back to a tiny error in the gene for hemoglobin, the vital protein in our red blood cells that carries oxygen. Normally, our red blood cells are round and flexible, easily navigating blood vessels. But in sickle cell disease, a mutation causes hemoglobin to clump together, forcing the red blood cells into a rigid, sticky sickle shape. These misshapen cells block blood flow like a traffic jam in tiny vessels. This oxygen starvation leads to devastating problems. Episodes of excruciating pain called VOCs, chronic anemia causing constant fatigue, and severe life-threatening complications over time like damage to vital organs including spleen, lungs, and kidneys, increased risk of infections and even strokes, sometimes occurring in childhood itself. Imagine living with that constant fear and limitation. For decades, treatment mainly focused on managing these symptoms with pain meds, antibiotics, and frequent blood transfusions. A true cure, like a bone marrow transplant, was out of reach for most due to donor matching difficulties and risks. Sickle cell often felt like a lifelong sentence until now. What if you could go into the DNA and fix that original typo, or at least bypass its effects? That's the promise of CRISPR. You have likely heard the buzzword CRISPR. It's a revolutionary gene editing technology inspired by how bacteria fights viruses, and so significant it earned its discoverers a Nobel Prize. Think of CRISPR Cas9 as an incredibly precise molecular scissor. It uses a guide RNA molecule like a GPS to find a very specific location in our DNA. Then, the Cas9 enzyme, the scissors, make a precise cut at that exact spot. This allows scientists to disable faulty genes or potentially make other corrections. It's like having a search and edit function for our genetic code. So, how do you use genetic scissors to fix sickle cell? By cleverly reactivating a healthy backup system we all possess. This brings us to a specific CRISPR gene therapy called Casgevy. Developed by Vertex Pharmaceuticals and CRISPR Therapeutics, Casgevy has a smart strategy. It doesn't directly fix the faulty adult hemoglobin gene. Instead, it focuses on restarting the production of healthy fetal hemoglobin, the type we all had before birth. Why? Because fetal hemoglobin works perfectly and doesn't cause red blood cells to sickle. The key is a gene called BCL11A, which acts like a switch that normally turns off fetal hemoglobin production after we are born. Casgevy uses CRISPR to find and disable this BCL11A switch in the patient's own blood stem cells. This process happens outside the body ex vivo. First, the patient's hematopoietic stem cells, the master cells in bone marrow that create all blood cells, are collected. Then, in a specialized lab, CRISPR is used to precisely edit these stem cells, turning off that BCL11A switch. This reprograms the cells to start making healthy fetal hemoglobin again. Meanwhile, the patient receives chemotherapy called conditioning to clear out their existing bone marrow, making space for edited cells. This is a tough but necessary step. Finally, the billions of newly edited stem cells are infused back into the patient's bloodstream. These powerful cells find their way to the bone marrow, settle in, and start building a new blood supply, one full of red blood cells carrying healthy, non-sickling fetal hemoglobin. The entire process from collection to recovery takes several months. So, does this high-tech reprogramming actually work? The results have been nothing short of breathtaking. In the landmark clinical trials for Casgevy, an incredible 93 to 100% of patients with severe sickle cell disease remain completely free from those agonizing pain crises for at least a year after treatment and often longer. Many who previously needed frequent blood transfusions became transfusion independent. Doctors saw significant increases in total hemoglobin and, crucially, high sustained levels of protective fetal hemoglobin. It's truly life-altering. Imagine hearing a patient say, "For the first time, I don't wake up fearing pain. I can plan my future. It's like being given a second chance at life." That's the impact. It's why many call Casgevy a functional cure, and making history in late 2023, Casgevy became one of the world's first ever approved therapies based on CRISPR technology, a monumental moment for medicine. This breakthrough opens doors. We have entered an era where precisely editing human DNA to treat disease is a reality. Casgevy is also approved for beta thalassemia, and research is rapidly advancing for CRISPR therapies targeting other genetic conditions like cystic fibrosis, Huntington's disease, and certain types of blindness and immune disorders. The potential is vast. But we must address the challenges. The biggest hurdle, the staggering cost, around $2.2 million per patient for Casgevy. This creates a major ethical dilemma about access and equity. How do we ensure these cures reach everyone who needs them, not just the wealthy? This is a critical global conversation. We also need to remember it's a complex procedure. The required chemotherapy has risk, including potential impacts on fertility and a small risk of later cancers. And because it's new, careful long-term monitoring is essential to track durability and safety, including minimizing any tiny risk of unintended off-target gene edits. So, we stand at a remarkable threshold. The power to edit our own genetic code is here. What are your thoughts on this incredible technology? Is CRISPR the ultimate game changer? How do we balance the benefits and the cost and ethical questions? Share your thoughtful opinions down in the comments below. Let's discuss this together. If this journey amazed you or gave you hope, please give this video a like, share it so more people can learn about this revolutionary science. And if you haven't already, please subscribe and hit the notification bell for more explorations into the future of science. The path ahead for gene therapy is both thrilling and complex. It promises a brighter future and demands wisdom and fairness. Thanks so much for watching today. I'll see you in the next video.